Hi, it's Dr. Phoenix. Today we're going to be looking at the cell cycle, or the series of events that cells go through as they grow and divide. In this picture, which animal would you say has the bigger cells? The obvious answer is that the elephant's cells are much bigger than the mouse cells. But the answer isn't obvious. The actual answer is that the cells in an elephant and the cells in a mouse are both the same size. They're both small. It's just that the elephant has many more cells. So cells are very small, and they have to stay small for a reason. And this is the reason why cells have to be so small. It has to do with the surface area to volume ratio. When we look at a cube, the surface area is all of the outside of the cube, whereas the volume would be all of the inside of the cube. So to determine the surface area, we take the dimensions of one side and then we multiply it by all 6 sides. So a 1 millimeter square, the surface area would be 1 times 1, which is 1 obviously, times 6 sides. So there would be 6 square millimeters of surface area. The volume of this cube would be length times width times height, 1 times 1 times 1, which would be 1 cubic millimeter. So the ratio is 6 to 1 surface area to volume in a small 1 millimeter cube. If we make this cube 2 millimeters, now the surface area is going to be 2 times 2, which is 4, times 6 sides, which is 24 square millimeters. The volume, again, is length times width times height, 2 times 2 times 2, which is going to be 8 cubic millimeters. Now our surface area to volume is 24 to 8, or 3 to 1. We can see that the surface area to volume ratio has dramatically dropped. There's much less surface area now. And if we double the cube again, we can see that the surface area, 4 times 4, 16 times 6 sides, would be 96 square millimeters. The volume would be 4 times 4 times 4, or 64 cubic millimeters. 96 over 64 is 1.5 to 1. We can see that the surface area to volume ratio continues to drop as the size of the cell increases. Well, how does a cube compare to a cell? And how does the surface area and the volume compare to a cell? The surface area would represent the actual cell membrane, or the surrounding of the cell. And the volume would represent what's inside the cell. So as the cell increases in size, the volume of the cell is increasing faster than the surface area is. There's much more volume in this cell than this cell compared to the surface area. So what does all that mean? Well, what happens through the surface area? What happens through the cell membrane? Things enter and leave the cell through the cell membrane. Nutrients, gases, waste products enter and leave through the cell membrane. So the things that the cell needs to survive on and get rid of pass through the cell membrane. Well, what goes on in the volume? All the activity of the cell goes on in the volume. All the chemical reactions, all the metabolic activity goes on in the volume. Well, how do things actually get through this cell membrane? If the white part of these cubes represents the cell, then the outer part of the white would be the cell membrane. Things get in and out of the cell through diffusion, and diffusion occurs only so fast. And so over a period of time, diffusion will occur at a rate that will allow molecules to enter from the surface of the cell all the way almost to the center of the cell when the cell is this small. It's going to penetrate approximately 9 millimeters into this cube. Well, diffusion only occurs at a certain rate. So if the cell gets bigger, diffusion is going to occur at the same speed. It's going to penetrate about 9 millimeters. So you can see that there's a large volume of this cell that's not going to get reached. And as we increase the size of the cell again, we can see, again, diffusion is going to reach about 9 millimeters into the cell. There's going to be an even greater area of the cell that's going to be unreached. So as the cell gets bigger, it's not capable of receiving the nutrients or getting rid of the waste products as efficiently as it should. So cells have to be maintained at a small size for that reason. And so before cells get too big, they'll divide. Other reasons why cells may divide is so that you can grow and develop. From the time you're a single-celled organism, a zygote, you're continuing to grow and you're continuing to develop. When cells are damaged or destroyed, they can be replaced by cell division. And some single-celled organisms will reproduce by dividing. So a cell goes through a life cycle. We call this the cell cycle, and there are two main stages. Those stages are interphase and cell division, the time when the cell is actually dividing. Interphase is actually 3 different phases or subphases. All right, the first subphase of interphase is called the G1 phase. The G1 phase. What goes on during the G1 phase is the cell's normal activity. A cell is doing what it's designed to do. So a liver cell is doing its liver functions, and a blood cell is doing what it's supposed to do, so on and so forth. And during its life cycle, it spends a great deal of time during this phase, doing what it's supposed to do. Then when a cell is getting ready to divide, it's going to go through the S phase. This is still part of interphase, and this is where the DNA in this cell is replicated. The DNA has to be copied so that the new cell has an exact copy of the DNA from the mother cell. And this happens during the S phase. S stands for synthesis or DNA synthesis. Finally, the third phase of interphase is called the G2 phase. The G2 phase is when the cell is getting prepared for mitosis. It's producing the enzymes and the structures that are needed for mitosis. The G1, the S, and the G2 phase. All right, the cell division actually is 2 stages. Mitosis is where the nucleus actually divides, and cytokinesis is when the cytoplasm divides. Mitosis and cytokinesis together are what make up cell division. So let's look at what goes on in these different phases. We've already looked at interphase. Interphase is when the cell is doing its normal cell activities. It's when the cell grows. That's the G1 phase. It's when DNA replicates during the S phase, and when it prepares for mitosis during the G2 phase. The next stage of mitosis is called prophase. Prophase, we're going to see the nuclear membrane starting to disappear. Now the chromosomes start to become visible. Right, up until now, the chromosomes were long and stretched out like a plate of spaghetti, and they're not really visible as individual entities. And so during interphase, the material is called chromatin. But now they begin to be called chromosomes when they become visible. And they take on this X shape. One arm of the X is called a chromatid, and the other arm is called its sister chromatid, and it's connected in the middle by a centromere. We can see that the centrioles are starting to move to opposite ends of the cell, and they're producing these fibers called spindle fibers. Eventually these spindle fibers will connect to each of the chromosomes. When they do, they're going to push the chromosomes toward the middle. And when the chromosomes reach the middle, we're now in metaphase. M in metaphase means middle. Metaphase, middle. The chromosomes are now all lined up in the middle. And then these spindle fibers are going to begin to pull, and they're going to separate these sister chromatids apart. And as soon as the sister chromatids separate, we then enter anaphase. Anaphase begins as soon as the sister chromatids separate, and then it continues as they are pulled apart toward the opposite poles. Once the daughter chromosomes, as they're called now, reach the opposite poles, we're now into telophase. Telophase, we're going to see the nuclear membranes are starting to reform. We're going to start to see the cytoplasm begin to pinch together, and that is called cytokinesis. Cytokinesis actually begins during telophase. Eventually the cytoplasm is going to completely pinch apart and form two daughter cells. Cytokinesis is now completed, and we have two daughter cells. The chromosomes are going to uncoil and get very long and thin and become known as chromatin again. And now we end up with two daughter cells that are now back in interphase, and they're going to restart a new cell cycle. Okay? All right. That is the cell cycle. I hope that was helpful. Feel free to bring your questions to class or send them to me on my big campus, and I'll answer them just as quickly as I can. All right. Have a great day, and I'll see you in class.